Hello, everyone, and welcome to today's webinar. Today we have three speakers, and we will be hosted by Professor Elizabeth Kellogg, who is a professor at the Donald Danforth Research Center. And I will go to the next slide. Today's focus is a celebration of the focus issue on the evolution of plant structure and function. It's the September 2022 issue, and it is edited by the four people you see here, all very well-known Evo Devo people. And it's a fantastic issue. It's online now, and I really encourage you to have a look. We'll hear more about this in just a moment. This webinar will be recorded and posted to our YouTube channel, the Plantae YouTube channel. You can find this and other recordings, and it's a great place to catch up on science and hear cutting edge research. I want to make a special thank you to ASPB member attendees. If you are not a member of ASPB, you can get 10% off your membership dues by using the code PRESENTS10, and the membership dues support this and other education and outreach projects we do. Finally, if you have questions for the speakers, please type them into the questions box, the Q&A box. If you have technical problems, you can email me at mwilliams@aspb.org. Or just try logging in again if you have difficulty. And so now I'm going to turn this over to our host, Elizabeth Kellogg, and I will stop sharing my screen.
Well, hello, everybody, and welcome. I'm glad you're here today to talk about plant structure and function and their evolution. This is a question, obviously, that has vexed biologists. It's made people curious for centuries, even before they called themselves biologists. It's a long-standing question as to how organisms got to look the way they are. But for a long time, it's a field that has been slow to develop, in part because it's actually a little challenging. And I remember a conversation with a colleague about 30 years ago, a very senior colleague, who said he really thought it was 30 years too early to be studying the evolution of plant development because the tools weren't available yet. So here we are 30 years later. And you can see from this focus issue, and you'll see from today's talks, that I think we've got the tools to start addressing many of these questions about how plants have come to be look so different. So if I can just share my screen here. Let's see if I can, and just think about this in a little more detail. Of course, what we're thinking about is the evolution of plants, and particularly with this focus issue, looking at how plants have evolved and diversified and just come to look so different. You can just look at the pictures across the top of this phylogeny, and they're just not the same. And so the question is, how did that happen? And of course, it's a combination. We end up studying in detail to answer that question, the phenotype, with better and better tools, and you'll be hearing about some of those in today's talks. Better and better phylogenies, and those will be anchoring the talks you're going to hear today, absolutely central to these questions. And then, of course, genetics, which also has become more detailed, more powerful, with more ways of looking at the genetic control of how plants put themselves together. In this focus issue, we ended up with 8 really terrific updates and several research articles, with more forthcoming, and these are going to be linked electronically to the focus issue, even if they're not in this particular September issue. The other ones will be added to it. So this is a really growing issue, just like it's a growing field, and there's also a research report. So if I can just go through and summarize a few of the updates, just introduce you to the titles of some of those. Several of them have to do with this, what we think of as sort of the first step, the colonization of the land, starting with a paper on plant microbe interactions, This is led by Camille Puginier, who's working with Pierre-Marc Delot. And Camille will be speaking today and be able to outline her work on this subject. Also a paper on apical growth, that is meristems and the single-celled versus multicellular meristems. And this is work by Jim Foraker and Jill Harrison. And work on transport tissues, the origin and evolution of what is called xylem in the vascular plants, but all the sort of xylem-related cell structures in other groups of plants, led by Sjoerd Woedenberg and Dolf Weijer. Papers on parallelism and diversification, where the similar things come up over and over, using some of the same toolkits, but putting the pieces together in a very different way each time. There's a paper on flowering time with Jill Preston and Siri Fjellheim, and Jill will be talking about this work today. And digestive systems of carnivorous plants, Mateusz Majda and Kenji Fukushima. I've come up in parallel multiple times, similar tools, similar structures, often slightly different results. And crassulation acid metabolism, paper by Carolina Hayduk, another pathway that has evolved multiple times in evolutionary history. And structural diversification. We have an update on the origin identity of the spikelet, led by Yanli Wang and Jincheng Zhong's group, and a paper on development and diversity in lignin patterns of Aurelia Emonet and Angela Hay. And Aurelia is here to talk to us today. So this is the group we have today. For moderating this session and leading the discussion, I'm going to turn it over to Mateusz Majda, who's just started working at the University of Lausanne, a lab working on biomechanics and the biophysical aspects of plant development. So Mateusz, yours.
Thank you, Toby, for your kind introduction, and thanks everyone for coming. Our first speaker of today is Aurelia Emonet. Aurelia obtained her PhD in 2020 from the University of Lausanne in Switzerland under supervision of Nico Geldner. Then she moved to Max Planck Institute for Plant Breeding Research in Cologne, Germany, and joined the group of Angela Hay as a postdoctoral researcher founded by Swiss National Science Foundation. She's interested in the evolution of lignin patterns and investigates mechanisms that control polar lignin deposition in the context of explosive seed dispersal in cardamina. With that, I welcome Aurelia to share her slides. And in the meantime, I would like to remind our audience to type your questions in Q&A chat box. Aurelia, I'm looking forward to your talk.
Thanks a lot. So let me just share everything. I think I need to go at the beginning, kind of a spoiler here. Yes. So first of all, thanks a lot for this kind of introduction, and thank you to the organizer of this webinar for giving me the opportunity to present the work I'm doing here in Cologne. So today I would like to talk a bit about development and diversity of lignin patterns. And I will focus on what I'm doing now in the group of Angela Hay, which is looking at lignin polarity in the context of seed dispersal. But before to go into these details, I just want to clarify what is lignin first. So lignin is a polymer of monolignol, which is present in the cell wall of the plants. And you can find it in the primary cell wall or in the secondary cell wall, mostly in the secondary one. And lignin is really a key component for evolution because first, this is the major constituent of foods, and it has driven actually the evolution of vascular plants. So putting lignin in the cell wall allowed the formation of conducting cells that were much more tough and that could transport water on longer distance, and this allowed the plant to colonize new environments. So lignin can be put in the cell wall in different patterns. And this pattern always goes with a specific function. So the most obvious example is the xylem, which are the water transporting cells. So in this xylem cell, lignin can be deposited in annular pattern or spiral pattern, but also to the most extreme in a pitted pattern when everything is covered with lignin except small dots. And this allows the water to flow through the cell because the lignin is quite hydrophobic, and these cells then conduct the water through the plants. Another good example of lignin deposition can be found in the roots. So it's the casparin strip you can find in the endodermis. So the endodermis is actually the cell layer that surrounds the vascular cylinder. So where all, for example, the xylem cells are found, or the phloem cells. And this endodermis has to be really tight to control what can goes in and out. And this is the role of the casparin strip that will form an apoplastic barrier between these endodermal cells so that it forms kind of a network around every single of these cells. Another quite nice example of lignin deposition can be found in the abscission zone. So when plants have to shed organs like the petal and sepal of the Arabidopsis flowers, then the deposition of lignin in the sepal and petal will allow the enzyme produced by the flowers to be stopped, so that the cutting of the petal and the sepal is really restricted to a very specific point. And this kind of form also a brace that keep every cells together. And a last example where it's this time a bit less developmental, but lignin can also be induced in response to immune response, and is thought to be able to restrict the spread of bacteria inside plants. But for my point of view, there is also sometimes where lignin can have striking pattern also outside of this kind of model plant I show you with Arabidopsis. So if we go a bit further, this is a plant of Cardamine hirsuta, so it's also in the Brassicaceae family. And these plants, they have an explosive dispersal. So that means the two valves of the fruit are able to separate and coil very fast to spread the seeds in a radius around 2 meters around the plant. And this specific pattern really rely on lignin deposition in the valve that form kind of a U shape with 3 rods and 2 thin hinges. And this deposition is quite different of what we found in other non-explosive plants like Arabidopsis, where the pattern is more forming like a box around the cell. This lignin deposition is actually found in the valve of the fruit. So here you have a fruit, and if you do a cross section, you can see in purple here the lignin. So this tissue layer, the endocarp, has this very specific U-shaped pattern. And to show you that this pattern is quite important to allow the explosion of the fruit, if you have a fruit, a mutant fruit, which do not possess this cell layer, the fruit is unable to explode. And reversely, if you put too much of lignin and in a way which is not as specific as this U shape, the fruit is also unable to explode. So that the pattern is really important for this specific trait of Cardamine. Moreover, this asymmetric lignin is always correlated with explosive pod shatter. So in the Cardamine genus, all fruits are able to explode, and they all possess this very specific U-shaped pattern. But in contrast, the rest of the Brassicaceae family, they have non-explosive fruits, and the pattern is this time also forming a box around the endocarp. So this is really like a black and white situation. And then to show that this polarization or this polar deposition of lignin is really important for the evolution of these traits. So to try to understand what kind of gene is important for the deposition of lignin in this polar fashion, for my project, I decided to use another plant, which is called Cardamine canopodifolia. So Cardamine is canopodifolia is also a Cardamine. He has explosive fruit, as you can see here. So for the moment, everything is normal. And also these fruit have a polar lignin deposition. But canopodifolia is also quite peculiar because it produces 2 types of fruits. The second type is actually growing directly into the soil. So here you see the fruit where the soil have been removed. And these subturned fruit, they are non-explosive. So we're in a case of anticopy where we have two kinds of fruit on the same plants. And if you look closer at these fruits, you can see that this time there is also lignin deposition, but they form a box shape, so they are non-polar. So this gives us a very nice opportunity to compare in a single plant two different structures and to understand what are the genes that regulate this lignin deposition. So we use Canopodifoliaceae, a new model system, develop it. So it has been sequenced, annotated, not yet annotated, sorry, assembled. And we do also some transcriptomic now to understand what is going on. Also, one thing I need to say is that this is an octaploid plant, so it makes things a little bit more difficult. So the idea we had was to compare subturned fruit with agar fruits. And what we did actually with this RNA-seq is that we took a valve of the fruits either just before lignification when there is not yet any lignin and during the process of lignification. And in that way, we do this for the subturned fruit and the agar fruits. And if we then compare before and during the process of lignification, we hope to find the genes which are important for lignin deposition. And then by comparing between the two types of fruits, we hope to find genes which are really specific for the polarity in these cells. So this is what we did, and we did it in two different ways. So we used Illumina sequencing to get quantifiable reads. And in parallel, we also use long read sequencing with PacBio to be able to have a reference transcriptome. And by combining the two together, hoping to find which genes are differentially expressed between this condition. And I show here you two heat maps because the first one is an alignment to the closest species, Hirsuta, and the second one is an alignment to our new transcriptome because we don't get the full genes with the transcriptome, but by using the full genome of Hirsuta, we were hoping to complete our analysis. So what you can see is that in this differentially expression for the interaction, there are quite a lot of different patterns. And to help you see a bit more what is going on here, so I will put some arrows where are the different samples. So the arrow goes from the less developed to the more developed states. And in blue, it's the agar fruit, and in yellow, this is the subturned fruit. So this goes like this. So you can see the clustering went a bit differently, but you can see the same kind of pattern between the two types of analyses. And by comparing these different clusters, we can find that some were very similar. And now, how to make sense of all this? So actually, we realized that there were some clusters that contain genes that we know are important for polarity of lignin. So this is work that have been previously published by a talented postdoc in our group, Miguel Perez-Anton, where he showed that lacases and peroxidases can also be polarly localized in Hirsuta. So these genes were found in this type of cluster where we have like a specific behavior where the genes are induced in the agar fruits during development, but downregulated or only very weakly upregulated in the subturned fruits. So by focusing on this cluster, I'm hoping to find genes that could be important for polarity and which are regulated in the same way than lacase and peroxidases. And what we found so far is that there were a lot of genes important for cell wall organization, regulation of symmetric cell division, pectin, also some hormone signaling, and also photosynthesis in response to light. Because of course we have like an agar fruits which have been grown in the light and a subturned fruit that have been grown in the soil. If we take this second cluster where the behavior is kind of the same, but much strongly induced in the agar fruits, we get also a lot of genes for lignin biosyntactic processes, also some for exocyst localization. So the exocyst is this system of membrane that will, this complex of protein that will put the vesicle at the right place in the cell, if we summarize a bit. And so a lot of candidates here. So we are now trying to find some that could be interesting, but we also decided to focus a bit more on lacases and peroxidases. So these genes I show that are polarly localized, they're important to activate the monolignol so that they can polymerize spontaneously. And these lacase in peroxidases, actually, we found them also in our analysis. So in orange is the one we've previously known that were important, but we found also other lacases and peroxidases that have the same pattern but have not been described in the context of explosive fruits. So this is some we expect could be also important for lignin polarity. And there are also another cluster of lacase and peroxidases which have opposite behavior. So they induce mostly in the subtern fruits and not and downregulating in the area fruits. And we think this one might be important to have more box-shaped pattern of lignin. So now we will try to localize this lacasein and peroxidases in Arabidopsis thaliana and Cardamina hirsuta in parallel to see if there is difference between the two species. I will finish with something a bit less related to lacases and peroxidases. So actually, when I was doing this RNA-seq, I was not so much happy with the fact that the area fruit and the subtern fruit were growing in very different conditions. So kind of worked hard to get a way to have subtern fruit growing outside of the soil. And so I managed to get some that were growing out, and they become actually green when you submit them to light. So that was one thing. But the thing that was the most interesting is that if you take these subtern fruits, you can actually observe that they explode. And when you look at the lignin pattern was completely different than the box shape we were expecting. So there is kind of a reverse of fates in these subtern fruits. So this gives us now a very nice tool to look a bit more into what can really regulate the lignin deposition. And this is a work we're doing with our master student, Karen Kelly. So we designed a new RNA-seq where this time we grow fruits, subtern fruits in a falcon tube that is covered with aluminum so that we can remove it to expose them to light. And the idea would be to then test after 30 minutes or 3 days when we're in the middle of the process of lignification, what are the genes which are induced. And in that way, we kind of remove all the genes which are maybe more important for like the shape of or the development of the fruit that do not interest us. So this is a work in progress, and we just get the RNA-seq results. So far, they are not yet analyzed. So we are really looking forward for that. So in summary, we show that Cardamina use explosive seed dispersal, and these rely on polar lignin deposition. This polar of lignin is always associated to explosive release. We developed Cardamina caudipodifolia as a new model system for comparative transcriptomic of explosive and non-explosive fruits. And finally, we found different genes enriched for cell wall, lignin, pectin, endomembrane system that could give us clue for what is important for polarity. And last but not least, light can trigger polar lignin deposition and explosiveness in non-explosive fruits. So with this, I would like to finish my talk, and thanks especially Angela for hosting me in her lab. Big thanks to Karen, which worked really hard in the dark for several hours dissecting valves, so he did a great job, and everyone in the lab for their help and inputs. And also to Bruno and Sonia that help us with the bioinformatics. So thanks a lot for your attention, and if you have any question, I will be happy to answer them.
Thank you, Aurelia, for a beautiful talk. Now we will move to the questions. So the first question is, could you please tell us more about the model organism you are using and the kinematics of dispersal? Is it the same like in Cardamina hirsuta, or it's a bit different?
So the plant we're using is so it's an octaploid plant, so it makes things slightly difficult. We can transform it, but it's also quite hard. So far we get one seed transform. But the process of the explosive dispersal is quite similar. So they both work in the same way with the tension, which is formed on one side, on the outside side of the valve, and the lignin is important to kind of restrict this tension. And when we get at the point where the lignin cannot stand it, then the structure of the lignin is really important to be able to release this energy. And this works really like a toy slab bracelets. Maybe if you see what it is, these bracelets, which are kind of very flat and a bit curved. And when you just crack them so that they become flatter, they can roll. So the valve is really working in the same way.
We have questions in the chat box. Please describe the role of lignin in abscission zone or in the flowers. So it's the question regarding if there is a role of lignin in the abscission zone in the flower.
Okay, in the flower soon. So in the abscission zone, you will find this lignin deposited in the petal and in the sepal. So the idea is that lignin will kind of let's say there is this abscission zone where that has to be cut. So on one side, the plant will produce enzyme to degrade the cell at that place, but the lignin will kind of avoid the enzyme to diffuse too freely around, so that they really stay at the place where they're supposed to degrade the cells. So in that way, they are kind of an apoplastic barrier to keep the enzyme at the same place. And then it is also thought that they kind of keep all the cells together like a brace to avoid them to go in to get detached and so on. So this is really something more in the sepal side and in the petal side than in the flower side. In the flower side, then to kind of seal the wound, it's mostly about cuticle formation, which is formed.
Here is a question, comment, very cool, especially the light induction part. Does the plant always make both types of roots? Are there conditions in which it might invest more in one or than the other type?
So in my hand, I always say it's doing both. There is some literature on canopodifolia because it has been already studied since 1930, where they show that actually if in stressful conditions, the plant will tend to use only subtone fruits in a way to kind of just invest in big seeds in a single place rather than trying to disperse and lose energy for that. So kind of staying at the place where probably the condition were good enough to have a plant growing there. But it has not been more testing than that. So there is a lot of things that can be done here.
We have many more questions, but maybe we can we'll just pick one of them. So how does lignin get localized inside the cell and how this is related to cytoskeleton? Okay,
so lignin, monolignin are produced inside the cell, and then they have to be put in the apoplasm, so in the cell wall. So either they could first freely diffuse, or it's also thought they can pass through a transporter. And then once they are inside the cell wall, they have to be polymerized. So this is where the lacase and the peroxidases are important. So they have to be at the right place. So the question is, all the peroxidase and lacase are deposited precisely at this place. We don't know it yet, but what we know so far, and also related to cytoskeleton, is that, at least in the case of the xylem, for example, the secondary cell wall and the lignin will be deposited in place where there is microtubule to drive the formation of the secondary cell wall. And these microtubule are depleted in places where there is no secondary cell wall and no lignin. So this is also a way to have like places with lignin and places without lignin. Now, exactly all these lacases and peroxidases are localized in a specific point. We don't know it yet.
Thank you once again for your talk. We still have many more questions, but we, due to the time constraints, will move to the second speaker of today. Our second speaker is Camille Puginier. Camille is a PhD student at the Plant Science Research Laboratory at the University of Toulouse in France. She's supervised by Jean Kiele and Pierre-Marc Dulot. During her master's, Camille studied plant science and plant microbe interactions, in which she used comparative phylogenomics to understand two symbioses, the arbuscular mycorrhizal symbiosis and lichens. For her PhD project, she aims to understand the molecular basis and evolution of lichenization in chlorophytes algae. And so for that, she combines phylogenomics, transcriptomics, and molecular biology. With that, I welcome Camille to share her slides. And in the meantime, I would like to encourage our audience to type the questions in the Q&A chat box. Camille, go ahead.
Hello. So hello everyone, and once again, thank you to Plant Physiology for giving me the opportunity to give this talk today. So I'm a PhD student at the Plant Science Research Laboratory in Toulouse. And I wrote a review about plant microbe interactions that have impacted plant terrestrialization with my PhD supervisors, Jean Keller and Pierre-Marc Delot. So the plants can be divided into two main groups that are the streptophytes and the chlorophytes algae, with the prasinodermophyte as a sister clade to both lineages. And within the streptophytes, we can find 6 algal clades, among which there is the Zygnematophysi class, that is considered as the sister clade to all embryophytes or the land plant that contains the flowering plants. So maybe the most impactful event of the streptophyte history was the colonization of the emerged land by the ancestor of embryophytes. However, terrestrial species can be found in other lineages of the green plants, including in algal clades, which means that several terrestrialization events might have happened and can be defined as successful and long-lasting wet to dry transitions. However, these wet to dry transitions comes with multiple challenges. For instance, solar radiations are not filtered by water anymore, which can lead to heat, highlights, and UV stresses. Secondly, the nutrient acquisition is not the same in aquatic and in land habitats, which can be a challenge as well. And finally, the water becomes limiting. And to overcome all these terrestrial lifestyle-related constraints, plant microbe mutualistic symbiosis are proposed as candidate terrestrialization innovations. So the first symbiosis that I'm going to talk about is the arbuscular mycorrhizal symbiosis that occurs between more than 85% of the land plants and glomeromycota fungi, also called arbuscular mycorrhizal fungi. And one of the challenges for mutualistic symbionts is to recognize each other. So this is why the arbuscular mycorrhizal symbiosis starts with a molecular dialogue involving keto-oligosaccharides that are released by the arbuscular mycorrhizal fungi and sensed by embryophytes. And embryophytes release strigolactone that can be sensed by the arbuscular mycorrhizal fungi. And recently, a work that was conducted in the team about the strigolactone biosynthesis gene called CCD8 showed that strigolactone have an ancestral function as symbiotic signals in both bryophytes and tracheophytes, which shows that the ability to form this arbuscular mycorrhizal symbiosis was acquired in the ancestor of all the land plants about 450 million years ago. So after this first step and molecular dialogue between the partner of the interaction, the arbuscular mycorrhizal fungi can invade the plant cells and develop structures that are called arbuscules. And these arbuscules are surrounded by the plant's plasma membrane, thus creating an optimized surface for nutrient exchanges. And for the plant, it allows the improvement of the water and nutrient uptake, thus helping the plants with one of the major terrestrial related constraints, that is the nutrient acquisition. And in the other way around, the lipid transfer from the plant to the arbuscular mycorrhizal fungi was studied in the team as well. And it was found that the lipid transfer is actually induced thanks to a transcription factor called wrinkle. And once again, this mechanism is conserved in all the land plants, showing once again that the ability to form this symbiosis was acquired in the ancestor of the land plants. And this symbiosis allowed a better nutrition for the plants that allowed them to conquer the land. So I just explained how the arbuscular mycorrhizal symbiosis appeared and allowed the colonization of land habitats. But there is another symbiosis that involves fungi and organisms from the green lineage, and especially some chlorophyte species that are algae, and they form lichens. So lichens are symbiotic organisms, as I said, composed of a microbiont or lichen-forming fungi that usually belong to the Ascomycota division and more rarely to the Basidiomycota division, and a photobiont that can either be a chlorophyte algae we called it lichen-forming algae, or it can be a cyanobacteria. And in this symbiosis, exchanges are made between the partners of the interaction. So the microbiont is provided with carbohydrates, and the photobiont is provided with water and minerals. But the microbiont is also responsible for the creation of a favorable microenvironment in which the photobiont is protected from desiccation and extreme solar radiation. So in the review, I decided to focus on the photobiont side, and more especially in green lineage organisms that are the chlorophytes algae. So within these chlorophytes, around 120 species are known to form lichens. But this capacity is restricted to two classes, the Trebuxiophyceae and the Ulvophyceae. However, in these classes, not all species are able to form lichens. And if we focus on the Ulvophyceae class, this capacity is restricted to two orders, the Ulvales and the Schwentipoleales. And once again, this capacity is, these orders do not contain species that are all involved in the lichenization process. So until now, we still don't know how to explain this distribution of the lichenization within the chlorophytes algae, which means that we don't know if the lichenization was acquired in the ancestor of both Trebuxiophyceae and Ulvophyceae, or and lost in the species that can no longer lichenize, or if it was acquired independently in Trebuxiophyceae and Ulvophyceae, which means in that case that it would be a convergent evolution. So this is why I started my PhD, because I wanted to understand how the lichenization evolved in chlorophytes algae. And by doing that, I hope to unravel the molecular mechanisms that are responsible for the establishment and the conservation of the symbiosis. And to answer these two tightly linked questions, I use comparative phylogenomics that consists in a whole genome comparison by taking the evolutionary context into account. And so thanks to this comparative phylogenomics approach, we were able to show that the ability to form lichens wasn't acquired in the ancestor of both Trebuxiophyceae and Ulvophyceae, which means that lichenization was at least gained once in Trebuxiophyceae and at least once in Ulvophyceae. And this result is actually coherent with previous studies of ancestral state reconstruction, and now we have the phylogenomic proof of this hypothesis. So now that we know a bit more about lichens evolution, we can dig a bit more on the mechanisms that are responsible for the symbiosis. And for that, we can use, as I said, the phylogenomic approach, but also the transcriptomic studies that were recently conducted. And to do that, the lichen-forming algae can be either cultured with or without their lichen-forming fungi, and thus we can identify the genes that are differentially expressed between these two conditions. And this is how we found some nutrient exchange genes that are upregulated during the symbiosis, such as ammonium and phosphate transporters that could be helpful for the photobiont, for the nutrient uptake, and thus helping them in these terrestrial habitats. But secondly, lichens are organized in layers, with the first one being the upper cortex, and is composed of compact fungal hyphae, under which we can find the algal layer within a region called the medulla, composed of loose fungal hyphae. And this upper cortex is responsible for the protection of the algal layer by protecting them from solar radiation-induced stresses such as UV heat and desiccation. But in addition to this physical barrier composed of fungal hyphae, we found that some gene families are expanded in lichen-forming algal genomes. And these expansions are associated with the stress tolerance. So for instance, we found a gene family in expansion involved in antioxidant enzymes or enzymes implicated in the redox homeostasis, We also found aquaporins and desiccation-related proteins that could be helpful for the lack of water in terrestrial habitats. So overall, it seems that the combination of the physical barrier composed of fungal hyphae and the expansion of gene family in lichen-forming algal genomes makes it possible for algal cells to live and thrive in terrestrial habitats and to be adapted to a wide range of environments. However, until now, there's still a big mystery concerning the lichen formation and how we can obtain such organisms inland. And recently, we found some clues about that, since an horizontal gene transfer of an enzyme called the glycoside hydrolase 8, or the GH8, might have shaped the evolution of the lichens in Trebuchet. As you can see here on this phylogenetic tree, the GH8 was transferred from bacteria, probably to lichen-forming algal cells. And since we didn't know anything about the GH8 biology in the lichen-forming algae, we decided to test the GH8 enzymatic activity to see what it could be degrading. And we found that the GH8 is responsible for the degradation of one type of carbohydrate that is called the lichenen. And the lichenen, as you may have guessed from its name, is a carbohydrate that is present in lichen-forming fungi hyphae. And to be more precise, the lichenen is present in the medulla region, as you can see here on this immunocytochemical localization. However, it was found that the lichenen is less abundant in fungal hyphae when in contact with lichen-forming algal cells, which is coherent with the fact that the algal GH8 is responsible for the fungal degradation of lichenen. And the hypothesis is that the GH8 is responsible for a better photobiont accommodation within the lichen thallus that thus allow them to be protected in this lichen organism. So to conclude, the terrestrial lifestyle comes with multiple challenges that can be overcome thanks to the association of plants with mutualistic symbionts. And I talked about the arbuscular mycorrhizal symbiosis that appeared approximately 450 million years ago. And this symbiosis was one of the reasons why plants were able to colonize the land habitats and to diversify. But plants also shaped the ecosystem in which other symbioses have evolved, such as the lichen symbiosis that appeared later in the evolution. So thank you for your attention. And once again, thank you to Plant Physiology and to my PhD supervisors, Jean Keller and Pierre-Marc Delot, and to all the EVO team in Toulouse.
Well, thank you, Camille, for an interesting talk.
Thank you.
So first question is, does the evolution always favor plants interacting with microbes, or it's not always the case?
So it depends if we talk about like pathogenic microbe or mutualistic microbes, but it seems that due to the fact that plants are cannot move, the interaction with organism is inevitable, and the fact that plant microbe interaction have been very widespread with mutualistic symbionts shows that it's truly one of the best way to accommodate to a lot of environments.
So to follow up, do you know how plants actually recognize those beneficial microbes and the parasitic ones?
Yeah, so as I said, in the case of the interaction with arbuscular mycorrhizal fungi, there's the molecular dialogue. Molecular signals are definitely one of the best way to recognize the good ones. And it is also possible with the lichen symbiosis, even though the studies are not very clear on that. But for example, the ribitol is one of the compound that is probably used as a molecular signal by lichen-forming algae and lichen-forming fungi. So this is the part for the good one, but of course, the receptors for bacterial effectors and so on can be a way to discriminate the good and the bad ones.
And if you look at the cell wall modifying enzymes, and there's the interaction with microbes, plant interaction with microbes, is there a kind of coevolution in the way plants modify their cell walls?
Yeah, sure. Actually, this is once again something that we can find in a lot of symbiosis. The plant modifying enzymes are actually responsible for this symbiotic interface that allow exchanges. We can see that in the arbuscular mycorrhizal symbiosis. We see it with the ectomycorrhizal symbiosis as well, and we see it as well in the lichen symbiosis. So this is a sort of convergent way to once again accommodate to the interaction with microbes that allow them to overcome a lot of constraints linked to mostly the nutrient acquisition in that case.
Here's the question about other kind of lichens. So what about the lichens that grow on the stems of trees? Is there any explanation why they grow on stems after terrestrialization?
Sorry, I didn't get the last part of the question.
So the question here is, what about the lichens that grow on stems of, like, you know, at the tops of the top of the tree? And is the question is there any explanation why they grow there after the process of going plants in the land, to the land?
Okay, so first of all, lichens can grow in different kinds of substrate. Like they can grow basically anywhere, like from cold to very hot and tropical ecosystems. So it's not, I think it's because they are adapted to a lot of environments that they can basically grow anywhere. So I don't think that the fact that they can grow on this type of substrate or this type of substrate is the question here, is that they can grow wherever they want, because they are adapted to a lot of stresses, as I mentioned, with a lot of enzymes and so on.
And the last question from the chat box is how you can use the knowledge about those genes involved in lichenization to apply to crop or economically important species.
So I mean, lichens are just very important species. They are considered as pioneer species at first. So I mean, understanding how they can adapt to, as I said, this wide range of environments can allow us to identify mechanisms that are implicated in, as I said, the adaptation and the tolerance of these stresses. So by identifying molecular function implicated in this tolerance might help us understand how other organisms can be adapted as well.
And then maybe the last one. So why lichens are growing much slower than plants?
Yeah, great question. It is actually one of the main limitation of the lichen study. Yeah, they grow very slowly, and I don't know if we can find a reason to that, but maybe the fact that they have no structure that can allow the acquisition, like roots, for example, in plants. Maybe it's the reason why they cannot grow as fast as plants, for example.
Thank you once again for your talk. And now we move to our last speaker of today. Our last speaker is Jill Preston. Jill is an associate professor of plant biology at the University of Vermont. In 2007, she received her PhD in ecology, evolution, and systematics from the University of Missouri St. Louis under the supervision of Elizabeth Kellogg. And then she did her postdoc at the University of Kansas with Lina Hilleman. Her lab studies the evolution of plant development with emphasis on how the economically important grass family has adapted to novel, often stressful environments shaped by past climate change. I'd like to turn it over now to Jill. And to our audience, please type your questions in the Q&A chat box.
Can you confirm you can see my screen okay?
Yep.
Great. Well, thanks very much for inviting me, and thanks for the introduction, and hello everybody. And today I'm going to talk about this part of the focus issue update that I wrote in collaboration with City Fjellheim on the evolution of temperature regulated flowering. So as most of us know, plants are really the fundamental source of food for all animals. And us humans derive most of our calories from the reproductive structures of plants, including things like seeds, grains, and fruits. So in order for plants to actually maximize their reproductive yield, they must flower when conditions are favorable. So this might be just during the summer for most temperate plants, so-called resting plants, but more broadly across the year for tropical plants, so-called non-resting plants. So if we focus on the temperate plants, we can think about flowering as really being a two-step process. The first step involves the transition from the vegetative juvenile phase to the vegetative adult phase. And this is a transition that basically readies plants to respond to inductive signals. So this usually involves winter signals, such as cold temperatures, which I'll refer to as vernalization, and short days. And then in the second step is basically the transition from vegetative to reproductive growth, which in turn is induced by warming temperatures and then the lengthening of the days. So clearly both high and low temperatures are important cues that might either promote or delay flowering depending on the environments to which plants are adapted. And we can think about as climate is rapidly changing under global warming, it's going to be unclear how different species will respond in their flowering time, because this will be affected both by the climate of origin and also by phylogeny. So with this in mind, City Fjellheim and I set out to review the literature on temperature regulated flowering. We focused on a number of questions, and I'm just going to talk about 3 of these questions today. So the first question seems very fundamental, but actually it's taken us a long time to get to grips with what the answer is, and we're still actually working on this. And that is, how do plants actually perceive different temperatures? Now, I'm not going to go into this in too much detail, but just sort of give you the take-home messages so far. So basically, the current literature suggests that there may be several different ways of sensing different temperatures, depending on the temperature range. With membrane stability being probably the most important for cold sensing, thermal reversion of phytochromes, which are known mostly as pigments that are involved in photoperiodic sensing, being important for ambient temperature sensing as well. And then heat shock proteins being important for high temperature sensing. Furthermore, there's also evidence that there might be different mechanisms important in different plant groups, with some overlap, of course. But one caveat to this is that we really haven't done a lot of consistent comparative studies in different groups. And so this might be something that changes in the future as we find maybe new mechanisms, but more overlap between these different groups. So the second major question I want to ask here is, how conserved are the cold, ambient, and high temperature flowering pathways across flowering plants? Now, this is a bit different to the perception of different temperatures, because here we're talking really about the transduction of that information actually into the plant and into plant development. So it turns out that it's something of an open question, this level of conservation for the ambient and high temperature pathways. There are definitely people working on this, but we don't know quite as much about these pathways. So I'll focus mostly on some of my work on the cold pathway. which is used in temperate species, again, to trigger flowering competency. And I'll just remind you that this pathway is also known as the vernalization pathway, and that's how I'm going to refer to it. So if we assume that vernalization responsiveness or vernalization responsive flowering evolved when global temperatures were cool, which happened only around about 36 million years ago, we can probably infer that that we have independent origins of this trait in clays that are older than 36 million years. So if we look at this angiosperm phylogeny, we can see that this response is actually found in many orders. So that's the little V in the yellow circles, because the orders themselves are older than 36 million years. And so we can infer from this type of reconstruction that it has probably evolved multiple times independently. Now, supporting these independent origins, evidence also shows that different genes have been recruited to vernalization responsive flowering, at least in the three major rosid, asterids, and monocotyledons. So, for example, in Arabidopsis thaliana, which is a rosid, one of the major regulators is flowering locus C, or FLC. In sugar beets, it's BTC1, and then in grasses, like wheat and barley, it's VRN1. So what about these patterns at narrower phylogenetic scales, such as within the grass family, which is what we've been focusing on? So we can ask, how labile is this trait? And again, are the same genes utilized? So focusing in on the grass families and the different subfamilies within the grasses, it looks like vernalization responsiveness has probably evolved, again, multiple times independently in these different subfamilies. So these arrows are basically showing at least 5 subfamilies into which vernalization responsive flowering has evolved. And again, this is based on the fact that these subfamilies are inferred to have diverged before the major global cooling event, around about 36 million years ago. So in looking at these, sort of comparing gene expression, genes that we know are important for vernalization response in some model species, we can then ask whether the same genes or different genes have been recruited to vernalization response in these subfamilies. And we've recently found, this is not yet published, but we've recently found evidence that yes, indeed, we have found multiple parallel recruitments of VRN1 and also its close parallel FUL2 in vernalization responsive flowering, at least within the Boideae, the Danthonioideae, and the Arundinoideae, with a couple of exceptions, the Chloridoideae and the Panicoideae. So far, we haven't found any evidence that either gene is important. So then let's zoom in on one of the subfamilies, the Boideae, which is what I mostly work on, that contains things like wheat and barley and oats. And this is actually the largest temperate subfamily of the grasses. And so you don't need to get into the weeds here and look at too many details, but basically we did a reconstruction of both vernalization responsive flowering, looking at various taxa across this subfamily, as well as looking at the response, the sort of the expression response in VRN1 across these different taxa. And what we found was there seems to be very good evidence of early recruitment of VRN1 at the base for vernalization responsive flowering. So in other words, we can see a very characteristic increase of VRN1 expression under vernalization conditions across vernalization responsive taxa in the subfamily. So it seems that vernalization responsive flowering was probably a really key trait for the expansion of these grasses into temperate regions. So our data suggests that vernalization responsive flowering evolved multiple times independently across angiosperms in response to cooling climates, and also that these plants recruited different genes to the traits at broad scales. but some of the same genes at more narrow phylogenetic scales. And then we do need, I should say, we do need some similar work to look at the ambient and the high temperature flowering pathways, and to look at the level of conservation and also the genes involved. So for the third question, third and final, we wanted to ask, what is the crosstalk between the high and the low temperature flowering pathways? So we actually know that in grasses, for example, vernalization is involved in modifying chromatin structure at the VRN1 locus to de-repress its expression. So basically, without cold, you start off having a VRN1 locus that has these repressive histone marks. And then with cold, you basically pull those repressive marks off and start putting active histone marks on, and so that VRN1 is now expressed. And then when VRN1 is turned on, it then turns on as a protein, it turns on another gene called VRN3 or flowering locus T, that eventually triggers flowering. Now, importantly, these active chromatin marks at the VRN1 locus are actually not removed when that vernalization period, that winter cold, is interrupted by warmish days. Okay, so in this regard, plants basically have this epigenetic memory of winter, and they know how much cold they've received, allowing them then to transition from the vegetative adult phase accordingly. And in fact, work in wheat and also a fairly close relative in the same subfamily, Brachypodium, has shown that giving an ambient temperature break in the middle of vernalization actually accentuates or accelerates the vernalization response. And so, as we can see in this graph, looking at flowering time, you can see that if you just vernalize without a break, this is the flowering time in terms of number of days. But if you give this break of 18 degrees, you actually see significantly earlier flowering. And so it's interesting to note that it's not just about cold, but it might be this relationship between cold and warmer temperatures. But on the other hand, if you give a break in the vernalization, a heat break, so sort of high temperatures around about 29 degrees, you can actually start repressing that VRN1 expression. So you start actually removing those active histone marks, and you get what we refer to as devernalization, or the loss of memory of winter. And so here you can see in this graph that under these very hot break conditions, you actually get this late flowering relative to straight through vernalization. And so it appears that flowering time in temperate plants can be fine-tuned not only by high and low temperatures per se, but also based on the order and the timing of these temperature cues. So understanding the complexities of temperature-regulated flowering is really critical to our food production, particularly given the likelihood of extreme future weather events, such as hot spells in the winter and then cold spells in the summer. And thanks for your attention. I'll take any questions.
Thank you very much, Jill. The first question is there any difference in vernalization response in T3 and T4 plants?
Sorry, I didn't hear the last part.
So the question is about the difference in vernalization between plants T3 and T4 plants.
Oh, yes. Yeah, so generally, I mean, you can get vernalization response in both, but within the grasses particularly, it tends to be the C3 plants that are in those temperate subfamilies. So you do tend to get more C3 plants showing the response than C4 plants, just because of that. But it doesn't necessarily mean they're kind of related to each other directly.
And another one, how long plants can stay in their vegetative adult phase, if you look at grasses?
Yeah, so for, it turns out kind of annoyingly quite a while if you're trying to get them to flower. So there's a lot of variation. And the length of the juvenile phase is actually pretty variable. And of course, it is different between, tends to be different between annuals and perennials, perennials having longer juvenile phases. But even when you get into the adult phase, you have to have inductive signals to actually induce flowering. And if plants don't receive those signals, they can remain in the vegetative phase for a long time.
And another is like how the difference in the temperature above ground and in the soil could influence the vernalization.
Yeah, so that's a really good question, particularly for grasses where their shoot apical meristem is generally kind of hidden, kind of, you know, at the soil level. So they can actually be protected a little bit from the cold, because they're at that level, and sometimes under snowpack as well. So when we do our experiments in the lab, it's a little bit artificial, because, you know, they're pretty much being party to the temperature of the air. But under normal conditions, yeah, you have these fluctuations in temperature, and the air is different from the soil. So that's one thing that we really need to know more about, is what happens under real life conditions, and as temperatures are kind of changing during the day and changing over, you know, over weeks and seasons.
There's still more interesting questions in the chat box, but we are running late, so I will conclude now. So by now we reached the end of our session. I would like to thank all our speakers, Toby, Mary, our audience for attending this seminar. Thank you all, and see you again.
Thank you. And we will post a recording of this in a few days. And we also have 2 webinars on circadian rhythms. Nice transition, Jill, coming up in October. So watch your emails. Thank you, speakers. Brilliant talks. Really enjoyed it. Bye-bye.
Thank you very much.
Thanks, everybody.
Thank you. Bye.
Bye.